Hello, and welcome to the Chapter 17 lecture on gene expression. You should use the information in this lecture to complete the Chapter 17 guided notes, which of course you should complete before you attend class. In the last chapter, you learned how DNA is put together, how it's stored inside cells, and how cells can make copies of the DNA. But we haven't really discussed the function of DNA yet. So what's it actually for? What does it do inside living cells? Well, DNA represents genetic information, so that means it stores information. What's that information for? Well, the information inside a gene, which is a short section of DNA, can be used to make other molecules, including RNAs and proteins. Both RNAs and proteins have other specific functions inside the cell. So the DNA is like a recipe book for building other functional molecules. The process of using the information in a piece of DNA to make a protein or an RNA product is called gene expression. So when a gene is turned on and is being used, we say that gene is being expressed. Now, proteins and RNAs represent the workforce of the cell. These molecules do all the little jobs that keep the cell alive and allow it to perform its function. Different cells are going to make different proteins at different times, and different types of cells are going to make different proteins than one another. For instance, a red blood cell contains hemoglobin. That's the protein that allows it to carry oxygen in the blood. But a muscle cell doesn't contain hemoglobin, it instead contains actin. That's a contractile protein that allows it to contract. This all relates back to a genetic basis. Because the information for making these molecules is stored in the DNA, by controlling which genes are turned on at which times, the cell can control its workforce and therefore control its functions. Gene expression consists of two processes, transcription and translation. Transcription occurs in the nucleus of eukaryotic cells. During this process, a gene will be read and a copy will be made of it. So that's what transcription literally means, is to make a copy. When I'm talking away up here making this lecture and you're writing some of that information down, you're actually transcribing what I'm saying. So in transcription, we make a copy. That copy is a piece of RNA. That transcript will then leave the nucleus and enter the process of translation. During translation, the piece of RNA will be taken in by a ribosome. The ribosome will read the information and use it to build an amino acid sequence, a protein. Now, during this process, because we're going from the language of nucleotides to the language of proteins, we're actually translating from one language to another. That's why it's referred to as translation. We can summarize this entire process by saying that DNA leads to RNA and RNA leads to protein. Sometimes this rule is referred to as the central dogma of molecular biology, because if you can control this process, you can essentially control the cell. To help you understand this overall process, I have this really silly analogy. Let's say that instead of a protein, that what you actually want to make is this delicious pie. Pies are kind of complicated to make, so you're going to need to approach this in two stages. First, you need to get the recipe. That process is called transcription. And second, you need to build the actual pie. That process is called translation. So first, let's get the recipe. Let's say that to get the recipe, you need to go to the library, which in this case is a nucleus, and you need to check out the book of recipes. The book that contains all of the recipes for all of the pies in the entire world is the DNA. Now that's a gigantic book and you don't want to carry it. So what you're going to do is make a Xerox copy of the specific recipe that you want. Inside the cell, this is going to be represented by a piece of mRNA, messenger RNA. Now that you have your Xerox, you can go outside of the nucleus and start translating this recipe into an actual pie. Now let's say that this recipe is particularly complicated and you need some help. So what you're going to do is find a chef to help you out. The chef represents the ribosome. What the ribosome does is he gathers up the ingredients and puts them together in the right combination to make the pie. That's translation. Now, the chef has a little assistant. It's a little molecule called a tRNA. And what the tRNA actually does is pick up specific ingredients and takes them to the chef in the right order so that the chef can make the pie. So at the end of this, you've got your amino acids put together in the right sequence. That's going to make a protein, or your finished product, the pie. The process of gene expression differs a little bit in prokaryotic versus eukaryotic cells. In prokaryotic cells, the process is much more straightforward and much simpler and much faster. Why is this? Well, remember that prokaryotic cells lack membrane-bound organelles, so all of the machinery of the cell is kind of thrown together in the cytoplasm. This means that as soon as a gene is transcribed, translation can immediately begin. So these guys tend to be really fast at expressing their genes. It also turns out that the genes of prokaryotic cells are a little bit more organized than those of eukaryotic cells. So again, the cell can access their genes a little faster and complete this process faster than eukaryotic cells. We'll talk about the reasons behind this in Chapter 18. Eukaryotic cells do contain membrane-bound organelles, so this means that transcription and translation occur in different places, and this slows the process down considerably. Transcription occurs on the DNA, so it occurs in the nucleus, and that's kept separate from translation. Once the transcript is made, it has to actually leave the nucleus through a pore, find a ribosome that's either going to be on the ER or floating out in the cytoplasm somewhere, and then translation can begin. So eukaryotic cells are not as fast at expressing their genes as prokaryotic cells. Here's a quick overview of how the genetic code works. We start with a nice piece of double-stranded DNA. Now, only one of the two strands of this DNA is going to hold the actual information, the actual gene. So in this case, let's say it's this bottom strand. What we want to do is make a copy of that bottom strand. We want to Xerox it or transcribe it. So to do that, we have to open up the two strands of DNA, and then we're going to add RNA nucleotides to the DNA so that it's complementary. So here we have a T in the DNA, we're going to match that with an A in the RNA. Here we have an A in the DNA, we're going to match that with a U, a uracil, because this is RNA. Remember, there are no T's in RNA. And so on. Now this little transcript that's been created, this piece of mRNA, can head out to translation. To translate it, what we're going to do is break it down into groups of 3. These groups of 3 are called codons. The cell is able to read each codon, it's like a word in the language, and it knows which amino acid it stands for. So the presence of that codon, AUG, tells the cell that it needs to put the amino acid methionine at the beginning of its protein. Next, we have this other codon, and that's going to tell the cell that it needs to put this other amino acid in that position. The third codon tells the cell which amino acid to put in the third position, and so on. Now, cells are able to decode the codons all by themselves, but how do we know which amino acids each codon stands for? Well, for that, we're going to use a handy-dandy chart. Let's say, for instance, that we have our first codon, AUG, and we want to know what amino acid that's going to translate into. To use this chart, I'm going to find my first letter A on the left-hand side of the chart. I'm going to look for, here's the A's. Next, I'm going to look for my second letter along the top, U. Here's U. That's going to lead me to this block. And then I can look in the block and find the codon I'm looking for, AUG. AUG codes for the amino acid methionine or MET. And yes, it is just fine to use the 3-letter abbreviations for the amino acids. You don't have to memorize this chart, and you don't have to memorize the names of all the amino acids. Next, we're going to examine the machinery that the cell uses to carry out gene expression. Several of the most important machines are made of RNA molecules. So just to remind you, RNA is single-stranded. That means that it has a single sugar phosphate backbone and that its bases just kind of stick out into space like this. Another important feature of RNA is that it uses the base uracil instead of thymine. So you'll see lots of letter U's in an RNA sequence, but you won't see the letter T. Keep that in mind. Because RNA is single-stranded and its bases just stick out into space, it means that RNA molecules can fold over on themselves and form three-dimensional shapes. Here, for instance, we have a piece of RNA that's folding on itself. And notice that these two little sections are complementary to one another. So they can actually grab one another and make hydrogen bonds, and that helps hold the shape together. Because RNAs can take on a three-dimensional shape, many of these molecules have specific jobs inside the cell in the same way that proteins with specific three-dimensional shapes have specific jobs. Three types of RNA are going to participate in gene expression. The first type is called messenger RNA, or mRNA. It actually carries the message from the DNA out to the process of translation. So this piece of mRNA is the actual Xerox that's made based on the recipe of DNA. Notice that it's just a long strip of RNA with its bases sticking out. It's not folded into a particular shape. Ribosomal RNA, or rRNA, is complexed together with other proteins to make the ribosome. The ribosome consists of two subunits, a large subunit and a small subunit that fit together. The ribosome, if you remember, is the chef in our analogy. So it's actually going to read the mRNA and it's going to use that information to build a protein. We'll see how it does this later. Transfer RNA is also known as tRNA. This is the little assistant from our previous analogy. As the name implies, tRNAs are going to transfer something. What they're going to transfer are specific amino acids over to the chef, to the ribosome, so that the ribosome can put them together in the right order to make the specific protein that we want to build. Now, it just so happens that tRNAs are T-shaped. That's coincidental, but it makes it a lot easier to remember. Transcription is the process of making a copy of a piece of DNA. Our copy machine in this process is this enzyme, RNA polymerase number 2, or RNA poly 2. What this enzyme complex does is it reads the piece of DNA we're interested in, and it uses that information to build the piece of RNA. It actually Xeroxes or copies that gene. Unlike the enzymes we discussed in DNA replication, RNA polymerase II is multifunctional. It performs all the jobs of transcription. It actually opens up the DNA by itself, makes the RNA copy by itself, and then seals the DNA back together at the end. So this process doesn't require helicase to open it. It doesn't require ligase to close it. RNA polymerase II does everything. There are three phases to transcription: initiation, elongation, and termination. Let's take a closer look at each phase. During initiation, the machinery of transcription is going to assemble at the beginning of the gene. It's going to assemble on an area that we call the promoter region. Now, the promoter region is a little section of DNA that doesn't code for anything. It includes a section called the TATA box that's just a series of thymines and adenines that repeat over and over and over again. This area provides a point of attachment for the machinery. So it provides something for the RNA polymerase number 2 to grab onto. Another thing to keep in mind is that even though DNA consists of two complementary strands, only one of those strands will contain the genes, the useful information. DNA polymerase number 2 will attach to both strands, and it will work along both strands, but it's only going to copy the useful strand, and it copies it from the 3' direction to the 5' direction. The strand on the other side is really just there to help hold that piece of DNA together. So it's a structural piece of DNA, but it doesn't contain any actual genes. The giant machine that assembles during initiation is called the Transcription Initiation Complex. This machine is going to assemble on the promoter. Here you can see the promoter with its little Tata box. The first components that assemble are molecules known as transcription factors. They actually help RNA polymerase number 2 grab onto the promoter, and it helps it open up that first little section of DNA. If any of the transcription factors are missing, transcription will not occur. So this entire machine has to assemble before you can enter the next phase of transcription. The second phase of transcription is known as elongation, in which the DNA will be read and the mRNA copy of the gene will be made. To accomplish this, RNA polymerase number 2 zips along the gene from the 3' end towards the 5' end. It opens up the DNA in a little bubble, and then it sucks in these free RNA nucleotides that are floating around, and it matches them with complementary DNA nucleotides. Here you can see a uracil being matched with an adenine. Here's an adenine being matched with a thymine. Here's a cytosine being matched with a guanine, and so on. As the nucleotides are added, the little strip of RNA that's formed will feed out of the back of this enzyme. During the third phase of transcription, which we call termination, RNA polymerase number 2 is going to hit the end of the gene, and at this point all of the machinery is going to fall apart. RNA polymerase will go off to find another gene to transcribe, the DNA will seal back up, and the little piece of completed RNA, which is called a transcript, will now go on to translation. Two different kinds of DNA sequences are used to end transcription. In eukaryotic cells, the machinery of the cell stops transcription once it hits the stop codon. But in prokaryotic cells, a section called a terminator sequence is used that usually occurs just beyond the stop codon.